Protein is usually presented as one of the foundations of healthy aging. It helps preserve muscle, supports recovery, and provides the raw materials needed to build enzymes, hormones, and tissues. Older adults are often encouraged to consume more of it to reduce muscle loss and frailty. But a new study has revealed a strange contradiction. When scientists reduced one essential amino acid in the diets of mice, the animals became leaner, maintained better blood sugar control, developed fewer signs of frailty, carried fewer senescent cells, and had a lower prevalence of cancer. Male mice also experienced a 23% increase in median lifespan. The amino acid was valine. Valine is one of the 9 essential amino acids that the human body cannot manufacture. It must be obtained through food. It is also one of 3 branched-chain amino acids, commonly known as BCAAs, along with leucine and isoleucine. BCAAs are especially popular in fitness culture because they participate in muscle metabolism and protein synthesis. They are naturally present in foods such as meat, dairy products, eggs, soy, beans, and many protein supplements. That makes the new result sound almost impossible. How could reducing an essential nutrient improve health and extend lifespan? The answer reveals why nutrition is more complicated than dividing foods into good and bad categories. In the study, published in Nature Aging in July 2026, researchers placed mice on carefully controlled diets beginning when the animals were approximately 1 month old. The control diet contained all 20 common amino acids and provided about 21% of its calories from protein. The experimental diet contained 67% less valine. Importantly, the researchers did not simply feed the animals less food. The diets contained the same levels of fat and carbohydrates, and the missing valine was replaced with non-essential amino acids so that total calories from amino acids remained unchanged. The valine-restricted mice were therefore not undergoing ordinary calorie restriction. In fact, relative to their body weight, they consumed more calories than the control animals. Yet they gained weight more slowly and accumulated less body fat. This suggests that changing the composition of protein altered how their bodies used energy. The valine-restricted mice showed increased energy expenditure. Their tissues appeared to waste more energy as heat through processes known as thermogenesis. Brown fat, a specialized tissue that burns fuel to produce heat, shifted away from storing lipids and toward greater energy use. The researchers also found signs that the muscles of male mice may have contributed to this increased heat production. This helped explain an apparent paradox. The animals ate generously but remained leaner because more of the energy was being burned rather than stored. Body weight, however, was only one part of the result. Both male and female mice maintained better glucose control across much of their lives. Male mice showed improved glucose tolerance beginning at a young age and continuing into old age. Female mice also showed improved glucose regulation, although some responses differed between the sexes. The liver appeared to handle fat more effectively. Valine-restricted mice accumulated smaller lipid droplets in the liver, while male mice carried less liver triglyceride. This pattern suggested that the liver was storing less excess fat and releasing more fuel for other tissues to use. These changes matter because impaired glucose control, insulin resistance, fatty liver, and excess visceral fat are strongly connected to age-related disease. But the most dramatic result appeared when the researchers followed the animals across their natural lifespans. Male mice consuming the valine-restricted diet lived longer. Their median lifespan increased by approximately 23%. Median lifespan refers to the age by which half the animals in a group have died. It is different from maximum lifespan, which describes the oldest individuals. An increase in median lifespan suggests that survival improved across a substantial portion of the group, rather than only in one exceptional animal. Female mice did not experience the same lifespan extension. This sex difference is one of the most important parts of the study. It shows why the result cannot be reduced to a simple claim that less valine makes all animals live longer. Both sexes gained several health benefits. They were leaner, showed improved glucose regulation, developed less frailty, carried fewer senescent cells, and had a lower prevalence of cancer. Female mice also displayed improvements in some measures of cognitive function and reductions in inflammatory brain cells. Yet only the males lived significantly longer. Scientists frequently observe sex-specific responses in aging studies. Male and female animals can differ in hormones, metabolism, immune function, fat distribution, gene activity, and disease patterns. A treatment that improves one biological pathway may therefore produce very different effects depending on sex. The molecular analysis in this experiment confirmed that difference. Researchers examined gene activity in the liver, muscle, and brown fat. The response to valine restriction varied dramatically between tissues and between males and females. The liver showed the strongest changes, with thousands of genes responding to the diet. Muscle and brown fat showed fewer changes, and the patterns often differed by sex. In male mice, the researchers identified a group of liver genes associated with mitochondrial metabolism. They also measured increased mitochondrial respiration. Mitochondria convert nutrients into usable cellular energy. Their decline is one of the major features associated with aging. Better mitochondrial respiration could help explain improved metabolic health and possibly the longer male lifespan. However, the study also produced a surprising finding. Branched-chain amino acids can activate a growth pathway known as mTORC1. Because excessive mTORC1 signaling is often linked to aging, researchers might expect valine restriction to reduce its activity. Instead, mTORC1 activity increased in the livers of the valine-restricted mice. This appears to conflict with one of the most familiar theories in longevity research. Drugs such as rapamycin extend lifespan in several animal models partly by inhibiting mTORC1. Protein restriction is also often believed to work by lowering this growth signal. The new study suggests that valine restriction may operate through a more complicated mechanism. Its benefits cannot be explained simply by switching mTORC1 off throughout the body. The biological effect may depend on timing, tissue, sex, mitochondrial activity, energy expenditure, and interactions among multiple pathways. This is an important reminder that nutrients are also signals. Valine does not only become part of a muscle protein. Its abundance informs cells about the nutritional environment. Changing that abundance may influence metabolism, gene activity, energy use, inflammation, and cellular growth. Previous research has found that restricting all three BCAAs can improve metabolic health and extend male mouse lifespan. Isoleucine restriction has also produced strong metabolic and longevity benefits in animal studies. Valine had received less attention as an individual target. The new experiment suggests that it has its own powerful effects, including reductions in frailty, cancer prevalence, and senescent cell burden. Senescent cells are damaged or stressed cells that have permanently stopped dividing. They can be useful during wound healing and cancer prevention, but when they accumulate with age, they release inflammatory signals that disturb surrounding tissues. Reducing their burden may help create a healthier tissue environment. However, this does not mean people should begin attempting to eliminate valine from their diets. Valine is essential. Humans require it for normal protein production, tissue repair, and metabolism. Severe deficiency could cause muscle loss, weakness, neurological problems, or other health consequences. The mice began the controlled diet at a very young age and remained on it throughout life. Every ingredient was measured precisely. This is completely different from a person removing several protein-rich foods while hoping to reproduce the same effect. The study was also performed in one inbred strain of laboratory mouse. Genetically diverse mice, other animal species, and humans may respond differently. A 23% increase in male mouse median lifespan cannot be translated directly into additional human years. Humans live in far more complicated environments. Our diets vary daily, and our outcomes are influenced by exercise, sleep, infections, medication, socioeconomic conditions, genetics, and hundreds of other factors. The sex difference also raises a warning. A nutritional intervention may improve certain biomarkers without extending life, and its effects may not be identical for men and women. Another concern is muscle health. The valine-restricted mice remained leaner and had a greater quadriceps mass relative to body weight, but they accumulated less absolute lean mass overall. In humans, especially older adults, excessive protein or amino acid restriction could increase the risk of sarcopenia. Any future strategy would need to balance metabolic benefits against the need to preserve muscle, bone, and physical function. That is why the most realistic outcome may not be a lifelong low-valine diet. Researchers could instead identify the molecular mechanisms activated by valine restriction and develop therapies that reproduce its benefits without causing deficiency. A drug might increase energy expenditure, improve mitochondrial respiration, reduce harmful senescent cells, or change liver metabolism while allowing people to maintain adequate nutrition. Scientists must first determine which pathways are responsible for the lifespan effect and why they worked only in male mice. Human research will also need to examine whether normal variation in valine consumption is related to metabolic health, frailty, cancer, and longevity. Short controlled trials could test whether modest reductions change insulin sensitivity or biological aging markers. Until then, the study should not be treated as a diet recommendation. Its real significance is conceptual. For decades, nutrition advice has focused mainly on total calories, carbohydrates, fat, and protein. But proteins are made from individual amino acids, and each may send different biological signals. Two diets containing the same number of calories and the same total amount of protein could affect aging differently because their amino acid composition is different. The experiment suggests that healthy aging may depend not only on how much protein we consume, but on the specific molecular instructions hidden inside it. Valine is essential for life. Yet in these mice, receiving substantially less of it changed metabolism across the body, reduced several signs of aging, and allowed males to live considerably longer. That result does not provide a longevity diet for humans. But it does reveal that one of the most ordinary building blocks in our food may also be quietly helping determine how quickly the body ages.